Okay, in this video we are going to briefly go over the steps of mRNA processing, which is something that has to happen to the strand of mRNA after transcription is done, but before the RNA leaves the nucleus. So this is kind of an in-between step between transcription and translation that will happen in the cytoplasm. This is a very important way that genes are regulated in eukaryotes because by processing the mRNA, you can actually change the type of protein that ultimately gets produced. Again, as we mentioned, it is going to occur before the mRNA leaves the nucleus. So a lot of people would just consider this to be a part of transcription since it happens in the nucleus with the rest of transcription. And processing itself is going to involve removing some segments of RNA and leaving the other ones behind, and also the addition of what we call a 5' or methyl cap on the 5' end, and this tail on the opposite end that is essentially just a bunch of adenine nucleotides, somewhere between 50 and 250 A's just added to the end of our mRNA strand. So let's look at those details, and on your Power Note sheet follow along with this diagram so that you can label everything appropriately. You have the exons are going to be segments of RNA that are kept in the strand. So those are the ones that are actually used for gene expression, meaning the sequence of those nucleotides is used to make a protein. That's why we call it an exon, because it is expressed. The exons in this diagram are shown in green, and you can tell that they are being kept because our final mRNA piece at the bottom has these green sections. Now those pink sections in between, we call those introns. Introns are intervening sequences, hence the name intron, and these are the sections that get removed. They are not used in the production of the protein, so they can be removed from our pre-mRNA and leave us with a final piece of RNA that only has the sequences that are needed for making the protein. Now, scientists aren't really sure about the origin or the purpose for a lot of these introns. That's still an area of very active research, but we do know that they're there and they do need to be removed from eukaryotic RNA before it can be used to make a protein. So let's take a look at the other two parts of your diagram. Number 3 is pointing to that poly A tail, which again is a bunch of adenine nucleotides added to the 3' end of our mRNA strand. And this helps to protect the RNA. The RNA is going to leave the nucleus and go out into the cytoplasm where there are enzymes called exonucleases. Exonuclease is an enzyme that degrades any kind of nucleic acid. So we don't want our RNA to get degraded because it's going to be used to make a protein. Therefore, we need to protect it, and that poly A tail will protect it because the enzymes will attack the tail, but it will leave the rest of the mRNA alone. And finally, the last part of mRNA processing is that methyl cap. And the methyl cap is on the 5' end of the mRNA strand. The methyl cap is actually added really early on in the process. It's not necessarily the last thing to be added, it's just the last thing that we are talking about in our list today. And the methyl cap, its purpose is to help the mRNA get where it needs to go. It helps transport the mRNA out of the nucleus and to the ribosome where it will be used to make a protein. So those were the steps of mRNA processing. Again, this happens right after transcription, but before the RNA leaves the nucleus, and it only happens in eukaryotes, and it allows them to regulate the expression of their genes. So hopefully this added information was helpful, and if you have any questions, please make sure you ask those in class.